We have an exceptional speaker today, Dr. Manu, and Andrew Manu will be our speaker today. First of all, I would like to thank Dr. Manu for and electrical conductivity meters as zoning tools for site-specific management of cold hardy grape and wine quality. A little bit of background about Dr. Manu. Dr. Manu from Iowa State University. He worked in Africa for 7 years and then joined Alabama A&M University as a full professor. In 2018, Dr. Manu joined the Department of Agronomy at ISU. In addition to his research, teaching, and outreach activities in the plant and soil sciences, he also serves as a George Washington Carver Endowed Chair and Professor in Charge of the Carver Scholarships and Academy that recognizes underrepresented groups in the United States. His main research interests include pedology, drone technologies, precision agriculture, and remote sensing. I am personally excited for Dr. Manu's speaker seminar because there are many opportunities we can gain from advancing drone technologies to advance our agriculture. To let everyone know, we will be recording our seminar. Dr. Manu, please, stage is all yours.
Thank you very much for that generous introduction. I was looking around to see if there's somebody else who we're talking about, but I think it's a pleasure to talk about some of the work we've been doing. But I'd like to acknowledge my collaborators, Mr. Joshua McDaniel, who is no more with Iowa State, and Dr. Ode Watrilo, who is in the Food and Nutrition department here at Iowa State. Okay. Now, it's a very short seminar, but I would like to give you the outline. I will try to talk about the relevance of this study at this time in Iowa, and then discuss a little bit about the concept of terroir. Just will be the literature part of it. And then we talk about how we characterize a vineyard here in Iowa. And then through veil zoning, then we did some soil characterization. Then also did a grape quality assessment. And then how to use all this approach to in the establishment of an integrative relationships between grape, soils, and the environment. Okay. Now, if anybody opens their palate and their eyes to looking at bottles and sniffing from bottles, we can say that Iowa is really advanced in terms of wine production. It's time that we do this kind of studies because the Iowan wines are thriving as tourism magnets and also as profit-making businesses. Now, if you look at statistics, in 2008, the number of wineries in Iowa was 77. In 2018, it moved to 113. And I'm sure I don't have the most current statistics, but I'm sure to be big, greater than this. If you look at the land in grapes, the grape acres were only 30 acres in 2000. Now it's 24, in 2014, it was 1,200. And again, if you look at 2021, it's going to be even bigger yet. The wine industry contributed $420 million to the state of Iowa in 2014. So again, we can go up, not down at all. Now, what makes Iowa wine even interesting is the Iowa wine trails that we have. There are 8 of these wine trails across the state with a lot of participating wineries. Now, What are these wine trails? These are where people go for hiking and going out there and tasting wine and having fun as recreation. And this has been very progressive, and you can see the map of the trail, and it keeps increasing. I'm sure it's going to increase some more. We have about 40 varieties of grapes in Iowa for making the wines, and that as to why we have these different wines in Iowa. But I'm going to just talk about these two, the Brianna. If you like Moscato wine, then Brianna is your friend. Then we can talk about the Frontenac, which is a red wine. If you like mellow, go for it. That is Frontenac. That's going to be your choice here. But we need to think about winemaking and vineyard establishment and winemaking as a concept, because it embraces the concept of terroir. And terroir is a French word that means land. So when this concept of terroir comes in, all it's saying is that this is integrated in the land, because the wine quality is very, very important to build culture. If the quality is bad, you're in trouble. So when we talk about terroir, is the final expression of the wine quality, which is a combined influence of soil, topography, climate, grape quality, variety, and cultural practices. So in non-scientific terms, all this is saying is that the concept of terroir implies that the wine produced in a given region is unique and cannot be duplicated elsewhere, even if the grapes and the techniques for making the wine are painstakingly duplicated. This is a very basic concept in winemaking. So wine produced in each given region is going to be unique. And even in Iowa, if you go from Northwest Iowa to Southwest Iowa, the wines are going to be different. Now, the component of terroir then people have worked a lot on it. There's a gentleman called Bromley in Australia. He's done a lot of work. And one of the things that they talk about are soil as a component of this terroir concept, because climate and all these things are involved. But when it comes to just the soils, Hilgari in 2000 said that deep calcareous soils with sandy loam textures and low fertility are the most suitable for vineyards. So when we talk about Iowa soils loaded with organic matter and so rich, in a way, they are saying this is not going to produce very good one. But then that's something up for debate. Bromley and Hamilton in 2007 also said that soils high in clay give high grape yield, but produce wines of low quality. And but wines of low quality is just low alcohol content. Then there are other sort of fertility parameters that who can also influence the wine quality. Number one, Bromley again said that if you have a lot of organic matter, it leads to the production of wines richer in nitrogen, but poor in quality. So now there's a trade-off here, as we talk about. Then if you keep on putting nitrogen in your soils, the vine vigor is going to be great. You see a lot of leaves and everything. The grapes are going to yield a lot, but the juices have few sugars, which means you're going to have less alcohol, and the phenolic compounds are also going to be lower. Again, two researchers, Constantini and Bruselli, in 2014, found that exchangeable potassium is highly correlated with the acidity of wine, which is something good. So you want to make sure that your field has enough potassium for this to happen. Trace elements, macronutrients. This is where I hear a lot of people talk about wine when it comes to macronutrients. Trace elements in appropriate quantities as one of the most important things that you get in wine. And it's very, very crucial that anytime people talk about soils and wine, macronutrients will come in. Now, iron, manganese, zinc, boron have all been found to be correlated with the grape juice properties, including the degree Brix, and also the levels of sugars, which are these important parameters, important properties that we use to judge the quality of wine. So if you can appropriately manage your macronutrients in the field, that's going to decrease grape acidity, and also in that sense will increase the wine quality. Okay. Now, the factor of terroir. Now, one of the things that kill vineyard operators is the significant within-field variability in the field, which also translates into variability in the grape quality. So they said, and they documented it, that grape attributes can vary from vine to vine, cluster to cluster, and even from berries to berry. Again, this phenomenon is likely the result of the interaction between the vine, the soil, and the climate, and of course, the management, how you manage the soil. Okay, so this serves as a motivation for this study that I'm going to report about. For us to use this intrinsic principle that soils are variable, and if they are variable, they are going to cause variability in grapes, and then it's going to also influence the wine we make. We have to make some decisions in understanding the soil variability, so we can make people who want to come into the wine industry, the vineyard industry, to make management decisions. And the decisions will be including, what kind of land do I use? What kind of varieties do I use? And what are the appropriate management systems that I bring to bear to make sure that I get the kind of wine I want and I get the market I want. Okay, so this is just the motivation that to exploit this intrinsic advantage of using the variability in the field, it will require an a priori mapping and characterization of the different zones within a vineyard, no matter how small it is, and no matter how big it is. We need to understand this variability within the field. Okay, so we set up studying the wine, and this study is carried out in Northwest Iowa, close to Sac City. The winery is called the Rustic River Winery. And I happened to, when I was running some travel courses some years ago, I went to one of our students here, Andrew Phillips is his name. And on the trip, he was talking about doing vineyards in Iowa. And at that time, I never thought there were any vineyards in Iowa. Well, he came back and he set up his own with his parents. And they have a 6-acre vineyard there. And so we went in there and said, could you work with us to investigate the integrative effect of within field vineyards, soil variability, and the quality of the cold hardy grape cultivars. This to me, after talking to the extension, wine extension agent here in Iowa, I found out that this could serve as a foundation study. And if we are successful, it can lead to a statewide initiative for maybe on every kind of soil type that we have here. So we established this grape and soil quality relationships along the Iowa Trail. And I think this is something that will be welcome because of the popularity of the Iowa Trail, Iowa Wine Trail. Okay, so this is where we did our work. This is the vineyard from above, and we are going to take a small tour on the vineyard here. And you can see we are going from two grape varieties that we have there, the Frontenac on your right-hand side of the screen, to the Brianna. And this covers an area of about 6 acres. I should let you know that actually that's the southern part because of the way we flew. But on the southern part, this is, and we can see that it's a younger plants because it's, they did it about 8 years ago compared to this one here that we were going to talk about. Okay, let me go back there. I don't need you anymore. So we went out there and took a survey of the soils in the vineyard, and we could identify 3 soil series. We had a flagler, which is mapped as having developed from alluvial parent material, and of course our famous Clarion, also mapped on the glacier till, and then the Wadena, which is an outwash derived soil. And this soil gives me some comfort because when we talk about parent materials in class Magram 182, the kids get a kick out of it because of the outwash parent material. To them is not something that looks real to them, but they have to accept it. I told them, so they have to believe it. Okay, so this is the setup of the vineyard there. You see the Frontenac in the red as a red wine, and Brianna as a white wine. We have the Saint Croix, but we did not use the Saint Croix at all. So we took 20 rows of vines, which is around 6 vines per row, 66 vines per row. And then 10 of them with red and 10 white. The first thing we did was to use our electromagnetic conductivity meter to do electroconductivity mapping. So we did the whole field with the EM, and we set the meter in a vertical orientation mode. So we could go down to 1.5 meters. So we took samples, these counts as we went along, and we took so many points of these conductivities to come up with a mapping scheme. So we put the conductivity readings in Arc Gis, and we use the Jenks natural breaks classification system to create 4 zones. And I can see this is a raw image on the screen there. And the purple areas are the people that are given very low readings. And then as you get greener, it gets stronger and stronger. So we have very low, medium, and high as our zones. Now what we did was to convert the raster into polygons so that we can get the 4 polygons. So we had to do some generalization to put some pixels in some specific zones. So as you see it has to be grouped somehow. If you look at the map, you see so many colors, but at the end of it all, we have these 4 zones: very low, low, medium, and high. And you can see the readings, the conductivity readings, the associated with those pixels. We went ahead and did some surface sampling. We sampled to 25 centimeters on a regular grid-based pattern, as you see on the screen. We took 20 samples, but I'm talking about 20 samples in this parcel. Beyond this, there's the south side. We did not use it. That's why I'm not showing it in this sampling scheme, but we had more points. But for this study, because of what happened, which I'll talk about later, we did not use them. So the samples were tested for texture, major macronutrients, and micronutrients. So let me share with you some results from the analysis that we got from these fields. Now, we started with the texture, and here I wanted to see, are the textures different for the different series? But it should be, of course, that's why they mapped the soil, but then we wanted to verify. We see some differences in the flagella, clarion, and wadena. Well, let me go on and show the other properties, and then I'll talk about the decision we made on this variability in the field there. The sand content is actually kind of opposite of the clay, of course. So this is also showing some variation or different means for the flagella, clarion, and wadena. Then we look at macronutrients, but I can't show all of it because we did a whole bunch, but this is the Bray one. You see a trend there where the flagella has more phosphorus than the clarion and then the wadena. Then we also look at potassium, and it's just a little, you cannot make any sense out of this kind of means that we have in there. Well, we look at our macronutrients, of course, and here the clarion seemed to be the king, reigning over the flagella and the wadena in terms of amounts. Then we also looked at iron, which showed the same trend in terms of the macronutrients. That was encouraging that at least those trends are following each other. Well, that was for we look at it based on the soils. Now you wanted to look at it based on the zones. So these are the kind of means that we have in the zone and the ranges and all associated with this box and whisker plots. So what we saw is that we need to be able to say something, then we have to go out there and do some ANOVA. So although these things are like this, we did not want to put our head and put up this phosphorus here and manganese here. But we find, this is the finding from this. The majority of the physical and chemical properties in the zones identified with DMI were not statistically different. In fact, if you look at just the raw means, they are different. But when we did the ANOVA, we realized they were not significantly different. Now, what was the problem? I think sample size was the problem. Because we had a poor vine establishment on the southern part of the vineyard, we do not have as many samples as we wanted. Now, as you can see on the southern part where I have the box, it's kind of really dry. And it's the vines are very, very not developing as fast as they should be. There's a problem there that needs to be solved by the farmers, Andy and the parents, because they did not produce any grapes at all throughout the year. So we had to abandon that. And of course, let me go back. Nature can also be cruel. We had a similar experiment set up at the Hort Research Station north of here. About 2 weeks before we got to the point where we could harvest anything, almighty derecho hit Iowa, and it flattened the vineyard. So we lost all these points that we could have put into the model to be able to make a decision or get some significance in these parameters. We did not, but that is what nature can do to you sometimes. So there's always the idea that soil tests do not actually do a very good job in telling the story about how vines get their nutrients. Because the soil might have the nutrient there, but due to some microclimate or macroenvironmental issues, the plants cannot take the nutrients. So people have found out that there's a poor relationship between the soil and plant nutrients. And this could be, as I said, due to within field variability in soil chemical and physical properties. Secondly, microclimate and micro-environmental conditions. These hamper the plant sometimes from taking up nutrients. However, petiole analysis is generally more useful and reliable for judging vine nutrient status than soil tests alone. You can combine the two and come up with a better management system than just relying on the soil analysis only. So we decided then that we can also try to do some sampling of the petioles. This is our zones. So we superimposed this map on it, where those lines are just areas within the management, the EM zones, where we decided to take petiole samples. Okay, so within this zone, we took about 75 to 100 petiole samples, and if we went to a vine, we took about 5 to 7 petioles per vine. We did this on the Brianna and the Frontenac. And then we analyzed the petioles for both macro and micronutrients. Let me share some of the results with you. Again, we are trying to see if these nutrients in the soil, in the petioles will reflect what we have in the zones, will reflect what we have in the soil type, and reflect what we have in the grape variety. Now, one of the things that I should say is that we already did the analysis on the soils, and we did not find those variabilities. So it did not surprise me that the nutrient levels in the petioles, the major nutrients, the zones was not a significant factor. The P was sometimes around 0.6, 0.9. So we did not want to think about it. The soil type also, they could not differentiate between the levels of the elements or the nutrients we are talking about. When we went to the micronutrients, still the same problem. and which is, again, maybe due to the sample size. However, when we looked at the grape variety, we realized a lot of differences. Calcium, sulfur, iron, manganese, copper, boron, zinc, they were all different for the Brianna and for the Frontenac. So we went ahead and did some mean separation to see how these things line up. So if you look at the Brianna, which is a white grape, versus the Frontenac, the P, the K, the calcium, they were all higher in the Frontenac than in the, and these are PTO analysis, please. The PTOs of the Frontenac than the Brianna. The same thing was done with the micronutrients, iron, manganese, boron, copper, and zinc. And the Frontenac always trumped the Brianna. So that was an interesting relationship that we found, which was not, maybe I wasn't expecting that, but that is what the results gave us, which was exciting. So now we wanted to look at some correlations between the PTO nutrients and the soil nutrients. So I'm going to look at P in the neutral versus the P in the, say in the soil. So I'll put this thing, a circle or maybe an oval on the screen here, trying to look at those correlation factors. You can see on that left there, where I'm comparing the amount of nutrients in the soil compared to what I have in the petioles. But the macronutrients, I do not see much of some relationships. So the petiole is not reflecting what is in the soil when it comes to the macronutrients. But when I look on this side of the table, I can see that the micronutrients, sulfur, zinc, manganese, iron, copper, and boron, they seem to have some relationship with what is in the soil. The macronutrients in the soil seem to have some very nice relationship with those in the petiole. Again, this is what most of this research is talking about, that maybe the petiole sampling would be a better approach than relying on the soil, because the soil, there could be a lot of very reasons why the plant cannot even take the nutrients. So this was a great finding that to corroborate whatever has been done in this kind of area of research on wine quality and grape quality. Okay, then we went into the drone, and this is my toy, okay? And so we take it to the field, and this is what we do. We play with toys, right? And I say, go out there and tell me something. Okay, we are using the EB. This is a fixed wing. Now, what is going every 10 seconds is going to take a picture. And so by the time it gets down on this 6 acres of land, it will take about 200 pictures. And these are just little pictures. So they have to be stitched together using E-motion. So it looks for tie points. What is similar in everything in this picture compared to that? And when it finds that, it just puts them together until it stitches all of them together to come up with an automosaic like this. This is the principle that why this one works. We like the EB because it's fully automated. You let it go, tell it go to Kanna's house, go and bring me some information, and you go and it brings it. You don't have to be in there. And also, I think the sensors on there are very, very, very sharp. So it costs a lot of money, but I think it's worth the investment. So when we got there, this information done, then we have to look at some vegetation indices that reflect the health of the crop. So we did 2 of these, and these are based on electromagnetic bands. So we did the most popular one, which is the normalized difference vegetation index, the NDVR, is basically taking the near infrared band and then taking out the red band and then dividing this by the sum of near infrared and the red. And you're going to get a value between -1 and 1. Then you also have which is very recent, the normal red edge index. Sensefly, a company, they went bonkers on this, and they came up with some interesting relationships that when you are looking at crops towards the end of the growth, the NDVI, the normal NDRE does a better job than, say, NDVI, because NDVI has a problem. When the canopy is very thick and very green, it tends to saturate. So if you get to 0.8, to get to 0.9 might not even happen because the thing is saturated. So they brought in the NDRE, or normal red edge index. This is calculated about the same, except that the end there, instead of using the red band, I'm now using the red edge band, which is the difference in this. So we did that for the vineyard, calculated these indices for the vineyard. But please note that we had to be careful because the vineyard had grass in the rows. And so if you are looking at these indices, you could be overwhelmed by the grass rather than looking at your vineyard. So what we did was we had to do some analysis to just obtain the indices for only the vine canopy. So we created a polyline shape file, which we imposed on the rows. So we had a buffer of 3 feet to be able to make sure that we are within the canopy of the vineyard of the vines, instead of looking at the grass in the rows. This was a great idea, and I think it helped us make some decisions on this. So this is, since we could isolate the vineyard, those canopies as in every zone, we were able to calculate the NDVI or develop the NDVIs and the NDRE for the zones. And we flew starting June 3rd, all the way every other week until August 13th. And this is how the evolution of the NDVI as time went on within the different zones. What got us jumping, excited, was the fact that there was a good distinction of the zones, although the soil properties did not favor us, but this relationship exists, that the NDVIs that we got was matching the zones appropriately. And this is what the NDRE also looked like, which is almost similar. So we could use any one of these to actually make a decision in terms of looking at the zones. We are saying, well, the medium zone matches what the NDV, the vegetation indices gave us. So we can say the zones are right in terms of the identification of these properties within it. Okay, so what we did then was to correlate the vegetation indices with the macronutrients in the PTO, because we really established right now that the macronutrients in the PTO is a better indicator of the nutrient level of these vineyards than what the soils gave us. So this is what we have. If you look at the sulfur, for example, see the high correlation between the sulfur content and the NDREs and even the NDVIs. We go to copper, we get the same thing. So looking at this, I can use this, the NDVI to, as a covariate in looking at the amount of these nutrients in the vineyard. So if a farmer can fly and develop these NDVIs, which right now they've come up with drones that you fly, and as you're flying, it's collecting data, they have a software on that is calculating the vegetation indices as it collects them. So it's very easy for them to see what relationship we have between these macronutrients and what we in the PTO and what the NDRE and NDVIs are telling us. That was really cool. Okay, but then the great kahuna, at the end of all, what are you looking for? You are looking at the grapes. Whether the soil is nutrients are there or something, you are looking at the grapes. So we had to do some grape sampling. So we went to the same zones that we used for the PTO sampling to collect some grapes. So we took these grapes in clusters, Brianna on the left, the front neck on the right. So we took these bunches from the clusters, and everything in that the bunch was put together. But when we were sampling, we had to make sure that we sample clusters that some will be above the canopy receiving sunlight, and those that are below that are blocked from the sunlight, because we wanted to make sure that we don't bias the system. We put this in a cooler, and then we bring it to the lab and we do some analysis. Now, the common grape quality parameters that we look at, first of all, we have to look at the grape weight and the berry diameter. Then we have to look at the soluble solids. And these sugars are the most important soluble solids. And this includes the glucose, the fructose, and this is related to the alcoholic degree of that grape. Then we also look at organic acids, mainly tartaric, malic, and citric acids. When you drink the wine and you feel some tart kind of taste, is due to these organic acids. We also looked at tannins. These tannins contribute to the mouthfeel and the color stability of red wine. So, you know, this will become more important than in the Frontenac than in the Brianna. We also look at the degree bricks or the sugar content. And this is actually based on this that the farmers harvest. They have these meters, that the handheld meters, they can test and see what kind of bricks I have before they sample. I think it's between 8 degrees and 20 degrees, then they would have. Then we also have to look at the pH of the grape that comes out of it. So these are the basic analysis we did on the grapes, and this was done by Dr. Watrilo in her lab at the Food and Nutrition Department. So this is just a table that I would like to share with you about what we got in terms of the Frontenac and the Brianna, because at this time, I'm not even looking at zones anymore. I'm not looking at the soils anymore. because most of these properties are not very different anyway, statistically, actually, I should say that. But if I look at all these parameters, the berry weight and the berry diameter, the Brianna trumps the Frontenac. And you can see from the picture that the Frontenac, the Brianna has bigger berries than the Frontenac. Everything else, the Frontenac was ahead when it comes to glucose, fructose, all the acids, and the pH was high, a little higher in Brianna than the Frontenac. Then the tetrable acidity, tannins, and everything. You can actually look at the tannin here, 223 in the Brianna, which was kind of different. So that means the Brianna has some properties more related to tannin than the Frontenac, which is where we had a different readings from them. So this gives, we're going to visiting with a farmer that was interesting to him, because he knows I do red wine, I do white wine, but these kind of analysis are something that he's very much interested in. But at least with this study, we've been able to set these two things apart. But that is not where we stopped. We wanted to know the correlation between the grape variety and soil micronutrients along in these different types, different parameters that we are looking at. So look at where, please, I have those circles. When it comes to berry weight, the correlation we got with zinc and sulfur in there. Then when we look at boron, it also has a high correlation with berry weight and the average diameter. The tannin is more related to, well, I won't say related, they move in the same direction. Tannins, iron, copper also do that. So in this, we are able to say that the macronutrients are something that we need to pay more attention to, because it's affecting the weight. If it's affecting the weight to get more juices, it's the tannin, the phenols, then these other acids are not being very much influenced, but copper is not also very bad in terms of the sugars. We got this also, this relationship. Okay, so in terms of final thoughts about this study, if you consider the current significance of the wine industry in Iowa, a statewide study is warranted, because farmers are getting excited. Some people are converting some of their lands into the cornfields into vineyards. But when we look at the economics of it with Andrew, it looks like it's worth it. It's worth it if they can do that. Not to the very grand scale, but at least if somebody has the time to do it, a few acres putting in vineyard will help also. Then we can also continue to explore the use of the US on-mine area system technology for the zonation, because we make an inroad into this. Most of the studies I've seen, they just do the NDVIs and things for the whole field, whole vineyard. Well, you have vineyards. It's not like corn, where it's just grown thick like that. You have all these spaces. We are able to segregate the vineyard, the vines from the grass. That is a great thing. Then we should go to the next step to get vintners involved so they can evaluate the wine quality for us. And here I always tell them, I have my skepticism about wine tasting, because they're going to test it and say, this is good or not. But I mean, the first one, maybe he's being very honest. About the fifth, sixth one, he's drunk. How can he even give me a result? Now, I'm sure they have means of doing that with machines and things like that. But my final word is that nature is powerful and can be destructive. These non-differences we are getting statistically is because we have this limited sample for the analysis. It's because the ratio came in and just blew us off the field. But it's still a good study, it's still good results, and I hope that we are trying to see if we can repeat the experiment this year. We have the soil analysis, we don't have to go there anymore. We just have to do the flying and then doing the grape and the epithelial analysis to see if we are going to get the same results or something different is also good. Well, thank you very much for your time, spending the last seminar and listening to me talk about wine. Thanks a lot.
Thank you, Dr. Manu. That was a great talk. Questions for Dr. Manu? Luis has a question. Yeah, Luis, go ahead and unmute yourself and you can ask your question.
Jolly.
Hello, Dr. Manu.
Yes,
Thank you for your presentation. It was very nice.
Thanks.
I just wanted to ask you, do you know, I don't know if the translation is okay, the graft holder? Do you understand what I mean?
No, sir.
Some native speaker knows what I'm trying to say?
The graft, sorry, where you graft the vines together?
No,
Yes.
the graft holder, I mean because you have one variety of grape,
Yes.
but what it's in the soil, it's a different part. So the plant is composed.
The grafted things.
Yes. So my question is, because you have two varieties, and I would like to know if this graft holder, or I don't know what is the name, it was the same.
Dr. McDaniel says rootstock. Is that the term you're looking for?
That's right.
Okay, rootstock.
Okay.
Perfect.
I think it's not grafted.
Okay.
It's not grafted. No, because even the lady Andy's mother told me about the history of this whole thing, where they got their vines, the grape from and everything. It's not grafted. He would have said she has history on everything.
Okay. No, because I grew up in a winery, and one of the things that we notice, it's even though you have the same variety, if you have a different type of graft, the wine quality was pretty different. So one way that we had to sort like the wines per quality was based on that. And I just wanted to say that.
Okay. Oh, thank you.
Thank you.
That's a great thing too.
Next, we have a question from Dr. McDaniel.
Okay.
Hey, Dr. Manu, thanks for the great presentation. I really enjoyed it.
Thank you.
I had a couple questions.
Yes.
Some of them, the last one might be a little philosophical, but the first one is a little more applied. I've actually been to that farm that you presented the data from, and I can't remember the name of the student that was there, but we took the freshman learning community field trip there.
That's Andrew.
Andrew, that's right. Yeah. So is Andrew going to use your data at all to manage his farm? Is he planning on using the data to inform, precision fertilizer management, or what are you planning on doing with the data and with him, and if he's getting access to it and gets to look at it? And then my second question is, it sounds like by the end of your presentation, you were at first really talking me up and believing in the idea of this terre noire, but at the end of it, you seem very skeptical, and it sounds like there's not really a terre noire in it, and maybe it's just based on fertilizer and management, and not really the soil so much that's providing it. Or maybe I read that wrong. Maybe you are not so skeptical of it and think that there is something to it. So I just want your thoughts on, after doing this study, and if you believe there's a terre noire and what's causing it.
Okay. Thank you for your question. It's a great question. Well, maybe if I sounded skeptical, I might have not. The problem I had is that it's not that the soil properties are not affecting the thing. We could actually even see some differences in there. I think it's all the matter of, if I have to make a statement, sample size is important. It's more important than we think. I think it's the properties are there showing some trends, but I cannot talk to you and say there are differences without saying prove it. And the prove it is that statistical significance. It did not come up. And I told you why I thought it's that way. But the terroir is important, because I know that if we go outside that field, and we are going to get different relationships there, because the soils might be completely different. The other one is about Andy and the mother. To be frank with you, Andy's mother is up on this thing more than we think. She has all the data on this wine over the years that she shared with us. So they are interested in getting this to see what they've been doing. And I think I'm supposed to visit with them. There is a COVID that kept us apart, but we had to go there to talk to them, because they are going to use it, especially with the macronutrients, because Madame told me about spraying some things before we flew. So I think she's thinking about it. So this will give her more ammunition to go out there and manage the vineyard as best as she can. Thanks.
Dr. Manu, we have another question, but I want to support your idea about the sample size. That sample size is really important, especially for 2K HST analysis. When I did my analysis for a similar project, and I saw that sample size was very good thing to show the significance between these properties. So I appreciated you said that. We have the question from Valeria. She says that, are there plans to do similar studies in other wineries?
Yeah, that's what you were trying to get some grant from the Iowa specialty crops. And it did not work out, but actually Dr. Lamke decided to support us to do that. So now we have to go to a different landform so that we don't sit on this Clarion Nicolet website thing, the glacial tail. So this we want to do that. But losing Josh is a big blow, and I'm still trying to pick up the pieces to see how I can do this again.
I also have a question. Dr. Manu, you mentioned that you evaluate the quality based on some trace elements. Are there indices developed to evaluate their sufficiency, like other major nutrients like NPK, especially for winery?
Well, right now with if like we said, we could not actually even see the uptake of the thing from the soil. Now, if we can do some regressions and tie those NDVIs to the nutrients, and maybe multiple regressions, then we can use it as a predictive tool. It's possible. And I think I read one paper like that, but they were not using drones and things like that, but they were using some handheld meters to do that.
Yeah, yeah. Thank you,
My pleasure.
everyone. Thank you so much for joining today. If there is not any more questions, it's been a great semester for soil science seminars. We are closing the soil science seminars for this semester. It's been a great pleasure to share the same environment with you all, and talk to you guys next semester.